Welcome everybody to the Image Processing and Image Analysis workshop. My name is Sebastian Rief and I will be your host for this session. This workshop will be split up into 3 parts of each roughly 30 minutes. Between the parts we will have breaks of 10 minutes in which you can ask questions. You can also ask questions in the chat during the sessions. Please ask them in the open chat for everyone so that everyone can profit from the answers given. During the last few days, everybody should have received an email containing a download link for the Geodik 2022 version. You should also have received a license for Geodik 2022 that should be valid until after the user meeting week. We will use this Geodik version and this license for this workshop. Additionally, you should have received an email containing a download link with the data required for this workshop. The zip file you downloaded contains the input data required to do this workshop and also the results that we will be producing. So if any point along the way you get lost due to some technical issues, there will be points where you can rejoin the process. Now a short introduction of myself. I studied mathematics and worked for 6 years in the Department of Mechanical Engineering at the University of Kaiserslautern before I joined Math the Market in 2018. I work as an application engineer in the support team and I write several of the user guides. Thus, if you have support requests, it is quite likely that you come into contact with me. In this workshop, we will tackle the topic of importing images into Geodik and processing them and also running some analysis on the segmented images. The goal at the end is that you are able to handle image data in Geodik, know how to import this, how to apply image filters and create segmentation of these images, and afterwards run analysis on the segmented images. For example, to obtain grain size distributions or grain diameter distributions. We will use 2 different datasets in this session. The first one is an NMC cathode out of a battery from the Battery Microstructure Library of the National Renewable Energy Laboratory of the USA. That scan was done by the University College London Chemical Engineering Department. The second one is an air filter media, such as you can purchase for a car, where we ordered a scan ourselves. Now let's get started with Geodik. Since this is the first workshop in this week, it's a good idea to start with the basics, and we will do this. And I will explain you now how to start Geodik for the first time and how to install your license file. I guess that all of you have installed Geodik already, and after this we can start it up. I do this here by double-clicking on my desktop icon, and well, then you will launch Geodik. The first message that many of you will see is the information that no license is installed. Some of you will already have installed it, but I will show for all the others how to do it here. You can just click OK. There's no other choice. And then in the next step, you have to choose the option to install a license file. The other options are not relevant here because you don't want to create a request. You just want to install the file. And then we use the Find License button here to browse to the license file. In my case, you can see it here already. I put the license file on my desktop, so I browse to the desktop and select the license file Geodik UM 2021 workshops.gelik. The gelik extension stands for Geodik license file, and well, the name with the G is just to differentiate it from other license files so that it's clear that this file belongs to Geodik. So we select it and then click on Open. Wait a moment until here all the information about the support period and so on is updated. Then you can also keep the option to set the license as default checked. And click OK. Accept the agreement. So that's usual that you have to accept the license agreement. And wait another moment until the information about the modules is download from the server. So first you get an information that the license is successfully installed. That's great. And now we have to wait another moment for the information about the modules which are available on the server. I think this might take some time on each of your computers too. So here is the Manage Floating License dialog. For now we don't have to do much here, but if you have a floating license installed in your company, you can select here which modules you want to log for your session and which you don't want to log. So if you have, for example, 2 licenses of Geodict, but only 1 license of FiberGeo, you can decide if you want to have it or if you want to keep it on the server for another of your colleagues. So now we have enough modules in this license here for all of you, so just click OK. You get another message that the license will expire on October 17th, which is 1 week later than the workshop ends, which should be fine for everyone. You can click OK, and now you're set to go in Geodict. So now we're here in the main user interface of Geodict. Here's a large empty area. This will be later used to display structures and volume fields. Then over here you have the information about the status and the current module. So the status shows information about your computer, like the CPU load or the memory usage. Then below you see, for example, your project folder and information about structures and objects if some are loaded. So since this is empty here, nothing is loaded right now. Below you see the currently active module. By default, if it is in your license, FiberGeo is always the default module in Geodict, so it is started at the beginning, and all the modules are look similar here in the user interface. So you can select options, like for example here, creating or piling or distributing fibers. Then you have an edit button to edit parameters for this option. For example, you can select here the size of your domain. And below you can access help for this module, which opens up the user interface. You can record a macro. This is grayed out because currently we're not recording a macro, but if you are recording a macro, you can record, for example, the generation process here. And then you have a button to start the computation, which is generated here because you generate a structure. In other cases, this button is called run to start the simulation. The first thing we will have to do is to set up our project folder. By default, it's always the folder My First Geodict Project in your Documents folder. But since all the data of your current Geodict session is saved into this project folder and you want to know where it is saved, it's always recommended to choose the project folder in the beginning. You can either select it by right-clicking here and click on Select Project Folder, or with the icon up here for the folder. So we click on that, click on Select Project Folder, and then you can see here the default is the My First Geodict Project, but we will change it. There is a project folder which we want to use to live it with the workshop data. In my case, I unpacked it on my desktop. So here's the folder Workshop EBR. And in there is a folder called Project Folder. We want to take that one. The folder Results M2M. This folder contains all the results that I created. So if you want to compare your results to my results, or if anything went wrong, you can find the data in there. But for now, we select the project folder here, double click it, and then make sure that project folder is marked below here, and click on Select Project Folder. So now everything is set correctly, and we want to start by importing a structure, by importing data from images. And for this, we will use the Import GeoVol module. So up here, click on Model, not on Model, click on Import and select Import GeoVol. click on that, and then you will see below here that the current module changes to import GeoVol. And then we only need to click the browse button to browse to the files that we want to load. Now your project folder opens, and in this folder we placed another folder which contains image files of a CT scan, which is called Cathode NMC 398NM. You can open this folder, and in there is a lot of image files, several hundreds for the slices of that structure, and you can select one of them. It doesn't matter which, so just select one of the files and click on open. Now the geometry import dialog opens, and you can choose several options. For example, you can directly choose to crop the data, which we won't do here because we want to do it later in the image processing in Geodict. Then it's important to make sure that the box for import all files and folder is checked. Therefore, we only need to select one file in the beginning, because then all the other files are selected automatically. And which is also important that we select the correct voxel length, because Geodict cannot know them from the image files alone. That's quite nice that the voxel length is written into the folder name. So you see here 398 nanometers. So we need to select the unit to nanometers, select NM here, and type in below here the 398, and then we are good to go, and click on image processing. This will take just some time until everything is loaded. And then the image processing dialog opens. For now, I want to hide it, because first I want to show you the visualization in Geodict directly. Because now we have something loaded, and it's more interesting to look at something than at nothing. So directly at the start, well, you see something black here. That's because we're here in the 2D view, and we see the first slice in the X direction of the structure, and well, there's a bit of black space around our sample, and well, therefore we don't really see anything here. To look more into the structure, you can, for example, use the slider here to choose other slices. So you can slice through there, and then you can directly see that you see more of the structure. But you will directly recognize that the contrast is not very good, so everything's very dark here. Therefore, we will directly change the color map here. We can do this by switching here over to the volume field tab. So the volume field tab is for volume fields, and image data from a CT scan is a volume field in Geodict. So you can switch over here, and then you can see the settings for the color map. And for this, we click on Edit. And as you can directly see in the histogram here, that there's a very wide range here, because somewhere will be some voxels here which will be a bit brighter, but the main part is only this small part here. So we can choose this here to put it nearer to there, and can close the dialog again, and you can directly see that everything got brighter and you see more of the structure. Well, yeah, then you can also here choose to see it from other sides, for example from the Z direction or the Y direction. But I think that's the main interesting things here about the 2D interface. Therefore, we directly change to the 3D interface. So up here, you can select 3D rendering instead of 2D. So click on that. And then it takes some time, and the volume field is then rendered in 3D. So here we are. And first you see the structure directly from the top. You can for example, rotate the structure by clicking it with the left mouse button and dragging the mouse. So here we can rotate the volume field. You can zoom either with the mouse wheel and steps, so you can see it here, or with the right mouse button. So click the right mouse button and drag it, and then you can click and hold the mouse wheel and drag it around here. Well, I think that's most about the how to rotate and handle this here. If you want to see, for example, inner part of the structure here, because there's a lot of black around here, that's not very interesting. You can clip inside of the volume field. So click here on the volume field tab, and then at the right you have the clip sliders, and then you can, for example, hold it here and drag it, and then you can clip inside of the volume field. You can do it on all the directions, so you can see a bit more inside of here. I think which might be also interesting is if you want to have something like a quick estimate of the thresholding on the structure. You can directly threshold a volume field in the visualization. So this doesn't really create a structure, but it's just to have a quick view. You can go over here to the threshold slider and click and drag it, and then the volume field is thresholded, and you can directly see where the bright spots are, and if you hit it at the right position, you can see that the grains start a bit to separate, and you can have a look inside of the structure. But now we want to go to the real image processing, therefore we just drag it back to 0 here and switch to the 3D image processing dialog. Here in the image processing dialog, you see directly the 3 areas with structure from the different views, so from Z direction, X direction, and Y direction. At the beginning you see the middle slice in all the directions, and I think one of the first things that you will see that everything looks red here, which is a bit weird, and this is because by default you get an overlay shown here of the segmentation, and since there's a lot of black area around here, the automatic segmentation only segments the outer area from the inner area, and therefore everything in there is shown in red. For now we don't want to see this overlay, and therefore we can change the visibility of the overlay up here. So just drag the slider down there, and well, you can change the visibility of the overlay and in the structure. So drag that down, and if you want, there are also other overlays available here, but for now we just keep the default of the segmentation. Then if you see the structure now better, I can show you the main controls here. So you can zoom in here by holding the control key and the mouse wheel. With the mouse wheel itself you can slide through the structure here. So just use the mouse wheel and scroll through there. The same holds here for all the other views. You can also zoom with the right mouse button and dragging, so you don't zoom in steps, but in smooth. And you can also pan with the sliders here at the side. So you can have a deeper look inside of the structure. You might directly see that, we already saw that we don't really need the black space around here. So the first thing we will do here is crop our structure. For this we go over here in the workflow. So you can either choose image processing or segmentation and labeling, and first we want to start with image processing to make our image ready for the segmentation. And here in list 3 we click on the image size subtree, so open that, and then select the crop option. In here, well, you can directly use the numbers here to crop the structure, and you directly see that there's a yellow line moving in here. You can see it here. And if you have a look up here, you can see that the overlay changed from segmentation to crop. So you can switch back to segmentation. Then you don't see the yellow line, and you can switch to crop, and you see that line here. This line indicates how the structure is cropped in this direction, and you can either use the sliders here, enter directly numbers, or you can also drag the lines here where you want them to have. So we just choose them to find a nice part inside of the structure, and the yellow lines are also faintly visible here at the side, so you can directly drag them somewhere where you want them to have. Sometimes they're a bit hard to grab. And well, I think this looks already quite nice in this direction, but we also have to make sure in the Z direction that we only get the area of interest. And you see here the areas are not very homogeneous, so we want to crop them out. So therefore we drag that down here, somewhere there, and also here. So I think this looks quite nice. Even with the overlay enabled, you can use the sliders to slice through the structure to see if it only contains the region of interest. I think this looks quite nice here, and also here. And then we only have a look at the numbers here to make that even numbers, because it's always a bit nicer. We change that here to 50 and to 750, and here we go from 200 to 700, and below here 225 to 485. So now we get a structure of 500 by 500 by 260, and to apply the crop, the only thing you have to do is just click on apply. It takes some time until the image processing is executed, and well, then you get the crop out here of the structure. So and what you might have already seen, that the contrast here is a lot better than it was initially than in Geodik directly. That's because the gray value range is automatically adapted, so that the areas at the borders are a bit cut out. Therefore it's a lot brighter here, and you can also have a look at the histogram down here to well, get a better estimate about the gray values in the structure. What we see here is that we now have a large area below here, which is practically not used, so from 0 to 1500, and well, we can also take that out of visualization, and we do this up here by selecting the gray value range. We see it goes, the selected gray value range goes until 12,032 here, which looks okay, I think. But I think we can choose another start point, perhaps around 2000 here. So just type in 2000 here, hit enter, and well, then the histogram looks a bit better here. Now, what you might also see is that well, everything looks a bit noisy here. That's because we want to filter our image here to get rid of the noise. But before we do the filtering, we want to go into the break. So if you already have questions about what I explained until now, before we go into the break, I will show you how to save the structure, or how to save the volume field. Since if you got lost during the process here, we can all start by loading the volume field anew and doing the filtering. So therefore, for saving, go up here to File. They can either choose to save a 2D image stack or save a 3D image. 2D image stacks create a lot of files and are usually larger and take longer to load. So therefore we save directly a 3D image. So click on there. And then we keep the default Geodict raw file, which is a raw file with a header from Geodict, which contains information as, for example, the voxel length. Then we select the compress option here to make the file a bit smaller and faster to write and load. And then we only need to click on Save 3D Image. And we call the file cathode nmc cropped. And just click on Save. Well, this takes some time. And now we go into the break. If you have questions, feel free to ask.